welcome to my YouTube channel. So without wasting our time, let's start with the next topic, that is packaging of DNA. See, what is packaging of DNA and why does it needed in our body? Obviously, if we going to calculate the total length of the DNA, it's approximately 2.2 meter for mammalian DNA. Okay, here I am talking about the mammalian DNA. The total length of mammalian DNA is 2.2 meter long, and whereas our cell is very minute, or you can say microscopic unit, we cannot see with the naked eye. Obviously we need the microscope, and instead of the cell we need to enter our DNA into the nucleus, obviously, which is very small as well. If the cell is like that, then the nucleus is only this structure, and in this structure we have to add on the 2.2 meter long DNA. It is the length of the single DNA that is present in our nucleus. Fine. Now, how to calculate the length of the DNA for the Cbse exam point of view? It is very important question, that how to calculate the length of the DNA. Okay. So the length calculated by the formula is the distance between two consecutive base pair with total number of base pair in a DNA. The distance between two consecutive, in case of the humans, the distance between two consecutive base pair is approximately 0.34 nanometer. But instead of the nanometer, we are going to take the unit into the meter. So we are going to convert it into the 0.34 nanometer multiplied by 10 raised to power 9 minus 9, then it would become 0.34 into 10 raised to power minus 9 meter. 1 nanometer is equal to minus 10 raised to power minus 9 meter. Okay, so instead of multiplying the unit with 0.34 nanometer, we are going to use the 0.34 into 10 raised to power 9 minus 9 meter. That is the distance between two consecutive base pairs, that is A and G. For example, the distance between these two consecutive base pair is 0.34 into 10 raised to power minus 9. And the total number of base pairs in the human DNA or mammalian DNA is, actually human DNA is 6.6 into 10 raised to power 9 base pair. How many? 6.6 into 10 raised to power 9 base pair. Then it would come out to be 2.2 meter. Okay. So this is the packaging of length of the DNA we need to calculate, and in packaging of DNA we need to add this 2.2 meter long DNA into the nucleus in case of the eukaryotes. Okay. Now packaging in prokaryote, if we talk about, it is very simple, since you know that the prokaryotic cells are not too much advanced, and we know it very well that in the prokaryotic cell nucleus is not there. So the DNA or the genetic material always reside in the cytoplasm, and the word we use for that is called as the nucleoid. Definitely from DNA, nucleoid is little bit different. Some modifications are always be there, and that modification is like DNA is our negatively charged molecule. Why it is negative charge? Because of the presence of the phosphate group. Okay, remember we have the three components in our DNA: sugar, nitrogenous bases, and phosphate. Phosphate provide the negative charge to my DNA. Okay, so this DNA need a positive charge protein. Some positive charge protein would be there, and that DNA, that is the double helical structure, and here it is the protein that is positive charge, DNA is negative charge. It definitely going to wound around, wrap around the positive charge DNA and form a condensed structure. Not too much condensed, I am not saying that it is too much condensed, it is little bit condensed. एक सिंगल हेलिकल स्ट्रक्चर से थोड़ा सा ज़्यादा ये कंडेंस हो जाता है, थोड़ा सा, then it is called as the nucleoid. That complete structure, Okay, not this single unit. I'm not saying that single unit is nucleoid, but the whole structure is now considered as the nucleoid in case of the prokaryotic cell. Fine. Now in case of the eukaryote, if I'm talking about, in eukaryote, obviously much advanced cell, so definitely the packaging is little bit advanced as comparative to prokaryotes. DNA is again same negative charge due to the presence of the phosphate group, which is common in case of prokaryote and eukaryote, and it also needs the positive charge protein, and that positive charge protein is called as, specifically name is given, that is called the histone protein. Since it is protein, so definitely made up of the DNA, amino acid. Because it is protein, तो प्रोटीन की सबसे छोटी यूनिट इट इज अमाइनो एसिड. डेफिनेटली आर हिस्टोन प्रोटीन इज ऑल्सो मेड अप ऑफ द अमाइनो एसिड्स, एंड द नेम ऑफ द अमाइनो एसिड्स इज, स्टूडेंट्स, इज इंपॉर्टेंट. यू हैव टू मार्क इट अंडरलाइन, और यू हैव टू राइट इट इन द बॉक्स, note box, that these are made up of the basic protein, and the name of those basic amino acids are lysine and arginine. MCQ की फॉर्म में that question can be asked. NCERT में this question is also be there, like the line was also given in the paragraph as well. Okay, so now this histone protein always lie in the form of the octamer. It has the 5 type. Octamer means 8. Okay, histone protein has 5 type: H1, H2, H3 and H4. Now H5, no H5. H2 is of 2 type: H2A and H2B. Fine. Total 1, 2, 3, 4, 5. 5 type of histone proteins are there. Now how the octamer is formed? It includes H2A, H2B, H3 and H4. This is 1 unit. 4 की बन गई 1 unit. Again with the same, it is again H2O, H2B, H3 and H4 again, and it also form the unit again 4 and 4, total 8. Thus it called as the octamer, histone octamer. H1 is used as clip to bind DNA with histone. हिस्टोन के साथ बाइंड करने के लिए हमें एक सपोर्ट की जरूरत पड़ती है, एंड दैट सपोर्ट इज प्रोवाइडेड बाय द H1 यूनिट. क्लियर? नाउ कम्स टू बी पैकेजिंग ऑफ DNA. इट इज वेरी सिंपल, yet need to understand. See, this is 1, 2, 3, 4. Fine. Suppose is it H2A, H2B, H3 and H4, and in the similar pattern 4 units are also again resides. मैं ये ऐसे बना रही हूँ so that it is possibly try to make you clear. This is also the octamer. Now on that, it's my DNA helical structure. It is double stranded DNA helical structure. It wound around it. Suppose ये आया मेरा DNA ऐसे. Okay, then I try to make it in a 3D form, but नहीं बन रहा थोड़ा सा, but कोई बात नहीं. हाथ भी लिया like this. Okay, यहाँ से निकला एक क्वेश्चन. This is my DNA, and to stabilize this DNA, to improve the binding, here the H1 protein is residing. H1 protein. Okay, the total length of this comes out to be 11 nanometer, and बच्चा लोग, we call it as the nucleosome. What is nucleosome? Histone octamer, 1 histone octamer plus DNA जब राउंड कर जाता है, देन वी कॉल इट एज द न्यूक्लियोसोम यूनिट. फाइन. So in this similar, nucleosome contain 200 base pair. One nucleosome contain 200 base pairs. ठीक है, कितना कंडेंस करना स्टार्ट कर दिया. Now suppose this is one new histone, this is second histone, this is third histone. Now on this DNA wound round in that way. Suppose like this. This is DNA, this is histone, and the collective unit is called nucleosome. Again, this is nucleosome 2, this is nucleosome 3, and the same way that forms a condensed structure, and that condensed structure, not too much condensed obviously, chromatin, which gives the appearance of bead on a string. जैसे एक मोतियों की माला होती है, that appearance is given by the DNA in the form of the chromatin, and that chromatin when condensed, highly condensed, to form chromosome. We all aware about the term chromosome, we all aware about the term DNA, but we get confused between DNA, chromatin and chromosome. DNA is singly or double helical structure. Chromatin consists of DNA plus protein, and that is specifically histone protein, and it condenses to form chromosome. Fine, during the cell division. Is it clear? Now this is nucleosome. I am drawing. ये मेरा DNA के साथ रैप कर गया, बना दिया न्यूक्लियोसोम हिस्टोन के ऊपर. सो यहाँ पर दैट लिंक पार्ट, दैट वी कॉल दी लिंकर DNA. तो ये लिंक कर रहा है एक न्यूक्लियोसोम को दूसरे न्यूक्लियोसोम के साथ, दैट वी कॉल लिंकर DNA. Clear? Yes, pretty much clear. Linker DNA is also there. Now the chromatin, it is of two types, heterochromatin and euchromatin. See, what is the difference between hetero and euchromatin? Euchromatin, one is transcriptionally inactive, where the other is transcriptionally active. It is tightly bound, it is loosely bound. Because it is tightly bound, it is darkly stained. If euchromatin is loosely bound, then it is light stained. And one important factor like this, we are drawing this. इस तरह से मैं अपना एक DNA बना रही हूँ, मैं वो हिस्टोन अभी बना देती हूँ. ये रहा हिस्टोन. You can see, जो tight area है that is hetero. Suppose this is the tighter area that we call heterochromatin, and the looser area is called the loosen area is euchromatin. Okay, I hope the concept is clear with the packaging of DNA. And if you like my channel, please like, share and subscribe. Thank you so much. All the best.